Let me ask you a simple question. What if a life-saving medicine contained a hidden impurity, so small, yet so dangerous, that it could trigger global drug recalls? That impurity is nitrosamine. In recent years, nitrosamine contamination has become one of the biggest challenges in pharmaceutical quality control. And detecting it requires highly advanced analytical techniques. In this presentation, I'll walk you through the top 5 nitrosamine testing methods you should know, including LC-MS, GC-MS, and more, and how they help protect patient safety worldwide. Nitrosamine testing is critical because these compounds are classified as probable human carcinogens. That means even extremely small amounts can pose serious health risks. Regulatory agencies like the FDA and EMA now require detection at ultra-low levels, often in parts per billion. To put that into perspective, that's like finding a single grain of sand in a truckload. Failure to detect nitrosamines accurately has already led to major drug recalls across the world. So testing is not just about compliance, it's about protecting patients and maintaining trust in medicines. Let's start with the gold standard of nitrosamine testing, LC-MS. This technique combines liquid chromatography with tandem mass spectrometry. First, liquid chromatography separates the nitrosamine from other components in the drug. Then, mass spectrometry detects and confirms it with very high precision. LC-MS offers extremely high sensitivity, allowing detection even below parts per billion levels. It is especially useful for polar and unstable nitrosamines that cannot tolerate high temperatures. Because of its accuracy and reliability, LC-MS is the most widely used method in pharmaceutical laboratories today. The second important method is GC-MS, or gas chromatography-mass spectrometry. This method is ideal for volatile nitrosamines like NDMA. Gas chromatography separates the compounds as they vaporize. Then, mass spectrometry identifies them based on their mass. GC-MS provides excellent peak separation and has been used successfully in many real-world drug recall investigations. However, it has one limitation. It cannot be used for thermally unstable nitrosamines because high temperatures may degrade them. So choosing GC-MS depends on the nature of the compound. Now let's look at an even more advanced version, GC-MS. This method uses 2 stages of mass filtering instead of 1. This extra filtering step removes background noise and improves accuracy significantly. As a result, GC-MS can detect nitrosamines at extremely low levels, even as low as 0.1 parts per billion. This level of sensitivity is essential for meeting strict regulatory requirements. It is especially valuable when confirming results and ensuring complete confidence in the data. Next is high-resolution mass spectrometry, or HRMS. This is one of the most powerful tools available today. HRMS measures mass with extremely high accuracy, often with sub-ppm error. This allows scientists to identify nitrosamines with exact precision. More importantly, HRMS can detect unknown or unexpected nitrosamines. This makes it critical for research, process safety, and degradation studies. Think of HRMS as a detective that can find hidden impurities no one was expecting. The fifth method is HPLC with UV detection. This method is often used for preliminary screening. It allows rapid initial checks and is relatively simple to perform. However, it has sensitivity limitations. Because nitrosamines exist at extremely low levels, HPLC UV alone may not detect them accurately. That's why it is mainly used for early screening, while confirmation is done using mass spectrometry methods. In real laboratory conditions, nitrosamine testing comes with several challenges. First, the drug matrix itself can interfere with detection. Excipients and active ingredients can mask the nitrosamine signal. Second, detection limits are extremely low, pushing instruments to their maximum capability. Third, sample preparation is critical. Improper extraction or cleanup can lead to inaccurate results. Finally, contamination can come from unexpected sources like reagents, solvents, or even glassware. This means analysts must work with extreme care and precision. Choosing the right testing method depends on several factors. If the nitrosamine is polar or thermally unstable, LC-MS-MS is the best choice. If the compound is volatile, GC-MS-MS works very well. If you are looking for unknown nitrosamines, HRMS is the most suitable method. Ultimately, the choice depends on the compound properties, sensitivity requirements, and regulatory expectations. Selecting the correct method ensures accurate and reliable results. At the end of the day, nitrosamine testing is not just about instruments or technology. It's about patient safety. By mastering these analytical methods, pharmaceutical scientists ensure the safety, quality, and integrity of medicines. These techniques help prevent harmful drug recalls and protect millions of lives. Because sometimes, detecting even the smallest impurity can make the biggest difference. Thank you.